the Predictive Safety Testing Consortium, or PSTC. We were the first consortium started at CPATH. We launched in 2006, and we're a collaboration that started when a group of toxicologists across pharmaceutical companies had similar data and wanted to combine that where they were seeing similar results and look for a way to gain regulatory acceptance for using new biomarkers to measure drug-induced kidney injury and be able to use those in IND studies in the future. Kidney biomarkers were the first organ that we focused on within PSTC because at the time and still, when detecting kidney injury in drug trials really doesn't happen until frequently you lose 40 to 60% of kidney function, and at that point you've lost a whole kidney function. And so these new drug development tools, these fluid biomarkers that we measure, are able to detect injury sooner than the traditional markers, as well as be more specific and be able to delineate different parts of the kidney that are being injured. Yeah, and building upon that first success that we had at PSTC, we launched into other organs and tissues that were either lacking in sensitivity or specificity to detect drug-induced organ injury early on. And so we have groups pursuing qualification at the FDA and the EMA for biomarkers to detect drug-induced liver injury, drug-induced vascular injury, drug-induced skeletal muscle injury, as well as drug-induced pancreatic injury. And we've had a few regulatory successes since our first qualification in 2008 and 2010. We have a clinical qualification of the kidney injury biomarkers in 2018, as well as receiving letters of support for our skeletal muscle injury biomarkers in 2014. And in 2021, we received a letter of support for a panel of pancreatic injury biomarkers, which was a first there for a safety biomarker, not measuring a protein, but actually measuring RNA, microRNA. Yeah, so we've always taken a translational approach within PSTC where we start off looking in animals and are able to match the injury measured from histology sections of the different organs and look at the changes in the fluid biomarkers and match the level of tissue injury to changes in biomarker concentrations in the blood. And being able to have that correlation in the animal and then moving into humans where we don't usually take biopsies of different tissues, but we can see the changes in the fluid biomarkers in either blood or urine.